Hello guys, my name is Dr. Varun and you are watching Chlorophyll classes only on YouTube. Well, this is for the 12th class and we're gonna be completing our chapter Biotechnology, Principle and Processes. Till now what we have done, we have completed the process, we have completed the tools and we have started with the process. So 4 steps has been completed. What were the 4 steps? Isolation of DNA, fragmentation of the DNA, for getting the gene of interest. Then we have even did that amplification of the gene of interest and the ligation. After ligation we have to put it inside the host body, and that is becomes the 5th step. So we're gonna be starting with it. Let's just start. Transferring of gene of interest into host cell, and this process is also called as gene transfer. Now this could be achieved by 2 different ways. One is a direct method, another is an indirect method. The direct method is that directly we putting that gene fragment inside the cell, or the other way is that with the help of a vector. So one is vectorless, other is vector procedure. Now the indirect would be what? Through vector or carrier, and direct would be what? Vectorless. So one by one we're going to be seeing the direct and the indirect method. Okay, so one by one we're going to be starting the first one, the indirect method. So we're going to be studying about bacteria, plant and animal, the different ways which are available to us. Okay, starting with the first, gene transfer in bacterial cell. Now here what happened, you have a bacteria. Let's take that this is the bacteria over us. Now what we will do, I am being the scientist and I want that this bacteria should take this DNA, recombinant DNA. So how I will make the bacteria do it? I will say that bacteria take it. Bacteria will like, I'm not gonna take it. So we said that like, okay, if you are not taking it, then I will put what? I will put this bacteria on a heat. Like for example, I will try to give it heat. So temperature will increase, bacteria will be like, bacteria still say that I won't take. No matter how much torture you will do, I won't take. We're like, okay, you don't torture, I will give next degree torture to you. After this heat, I will put it into the ice. So it's like, ice. So now what will happen? Certain bacteria might take it, certain bacteria still not taking it. So I did it what? That I will put it again on the heat. So torture I will be giving. So it is called a heat shock method. And by that it try to take it. Even I put it what? I will put it on the divalent calcium ions also. Okay, means calcium 2 positive ions, because it will make porous. Okay, it will make the membrane more susceptible to take the things. Understood this thing? So this is how I will make bacteria, and bacteria might finally will take it. Not all the bacteria, certain bacteria will take it, but certain of the students who don't listen to anyone, Okay, they are not able to take it. So later on we are able to find it out also, which take and which not. Like same way, that would be a step. So we will learn it over there. Okay, so let's write about it. So what is that? Since DNA is hydrophilic, so it can't pass through the cell membrane, which is hydrophobic, because it is made up of what? Lipids. Okay, hydrophilic molecule. It cannot pass through. It cannot pass through cell membrane. So we have force the bacteria to take it. Okay, so we force bacteria on, or force bacteria to take up the plasmid. Take up the plasmid. Plasmid, which one? Recombinant DNA one. Recombinant one. Okay, not the normal plasmid. Recombinant, by making it competent to take DNA. Then moving on, how we will take it? We put bacteria on ice, followed by 42 degrees Celsius heat, and this is called as heat shock experiment or the heat shock treatment, which I call it what? Third degree treatment. Okay, and again putting on ice. Okay, then what will happen? Along with treating them with divalent calcium ions. This is only I have told you that. So these things you have to remember about that, and the bacteria, few bacteria will take it up, few will not take it up. There what? Your head teacher or the PT teacher will come and find it out who are not in their uniforms. Okay, so we will learn that how they will do it later. Moving on, this is about the bacteria. But we have a plant cells also. So plant cells, we have a little different opportunities. Okay, so second is gene transfer in plant cells. Now in this, what happened? Imagine that there is a bacteriophage which is able to go, or there is a bacteria, okay, which is able to give its genetic material or give a particular plasmid inside the cell naturally. That happens actually in case of plants. So we use a bacteria which is what? Agrobacterium tunificans. This bacteria has a plasmid. Its plasmid name is TI plasmid because of the presence of tumor inducing genes inside it. So TI is what? Tumor inducing genes. Now what does tumor inducing genes do? They actually, whenever a bacteria, for example, I am the plant standing like this, and a bacteria came and it goes and infect my cell, what it will do? That inside my cell it will release its plasmid, and as it will release its plasmid, what that plasmid has? It has tumor inducing capabilities. Tumors is what? Tumor is rapid cell growth will take place and creates what? A bulging tumor kind of a structure over there, right? Easy way to remember, basic layman language. Okay, so now what happened? That tumor has been formed inside on my body cells, different body parts, wherever the infection is there, they formed it. Means what happened? That naturally there is a DNA plasmid that is going inside the cell of that. Okay, this capability we can use it. What about the tumor that causing? So why not we cut the tumor cell and there only we put the gene of interest? What will happen? Now here comes a very important factor which will come up. Now think about something. If you have made a line, okay, it is a red color line, and if I cut it and add another thread of blue color, what will happen? The red color integrity has lost. Same thing happens over there. When you will cut a gene with a restriction endonuclease, its basic characteristic is lost because it works together. And how we will remember that? We have studied about the molecular basis of inheritance. In molecular basis of inheritance, we know that how the protein is being synthesized, because basically why we are doing this, so that it can show the expression of the protein, the gene, specific genes which we want. But we have cut it the tumor inducing gene, which means it lost the feature of tumor inducing, and over there I have added what? I have 100% with surety added what? My fragment. So that's how we can work it up. Understood this thing? Same way we have root hair inducing, hairy root inducing plasmids also present, which do what? Which make the roots hairy. But if we'll cut that and include our gene of interest over there 100%ly confirmatory, then what will happen? Our gene will express, not the other one. And this is how we are able to find it also, that if the tumor is not happening, if the hairy roots not happening after getting the plasmid, means our recombinant technology has been preferably done. And this will come back later on for your understanding. I hope you would have understood. We will come back to these things again. Okay. So in plants, what happened? A plant pathogenic bacterium, the name is what? Agrobacterium tumefaciens. Agrobacterium tumefaciens produce, what it produce? It produce plant tumor in dicot plant. So means it is specific to the dicot plants only. Okay, dicot plants, as it has plasmid, means this agrobacterium has a plasmid which forms tumors, and we call it what? Ti plasmid. Understood? Now this Ti means tumor inducing plasmid. Okay, tumor inducing. Then agrobacterium naturally has this capacity of transferring it to the plant cell, right? Agrobacterium, don't forget to underline this agrobacterium and tumefaciens separately whenever you are writing it, because these are what? Scientific name. Your marks will be deducted in your board examination if you will not do it. So it is a tip for the board's examination. Naturally transfer some part of Ti plasmid into host without any human efforts. So it is called as natural. It is called as natural genetic engineer of plant. This concept is understood. Okay, Ti plasmid has two parts. Ti plasmid has two parts. What are the two parts? Number one is the tDNA and one is wire region. So what is happening over here? This that we're going to see. So inside the host plant cell, tDNA is separated from Ti plasmid and integrate into host plant DNA and causes tumor. So we understood what that T will do what? It will cut from the plasmid itself and go and attach to the DNA of the plant cell. This to the T. What about the word reason? Okay, so word reason contains genes which are essential for tDNA transfer and integration to host plant DNA. So when we have to use this, what we will do? We will remove this tumor inducing thing. Okay. So when we use Ti plasmid as vector, we remove the tumor causing genes from tDNA region, because rest all of the thing that should go and integrate with the plant DNA itself, we need it. Okay, so then the desired gene of interest is inserted in place of it. Now the plasmid is called disarmed plasmid, because it do not have its weapon of causing tumor. Same Ri plasmids is there. Okay, so here we're going to be writing it up. Just same Ri plasmid of Agrobacterium rhizogens causing hairy root disease in plants. So we understood that how the plants do it. Let's go to the animal ones. In case of animals, what happened? We have our like retroviruses, those viruses which has a transduction as a method of giving the gene of interest, which we have even studied in the Hershey and Chase experiment, which clearly proved that DNA is a genetic material, right? So that thing will come over here. That concept is over here as well. So the third one is gene transfer in animals. So we have what? Retroviruses have also been disarmed and now used to deliver desirable genes into animal cells. Now coming back, this is the indirect method. Now comes the direct method. Straightforward, the name, and you have to remember what is happening over there. Okay, so the second one was direct method. Or it is called as what? Vectorless method. In this, number one is electroporation. In this, pore means electroporation means pores will be created, and what will be happening on the cell membrane. Okay, there will be certain pores, temporary pores we will create it with the help of electricity, and through that we will directly inject what, our DNA, gene of interest. Okay, so what will happen, it is what, creating temporary pores in plasma membrane. Next, let's see chemicals. So there are certain chemicals like polyethylene glycol. We will give that, it easily uptake the DNA with itself and goes inside the cell. Okay, so next is polyethylene glycol, PEG, helps in uptake of foreign DNA. So means it makes it more competent to take the DNA. Then third is micro injections. Name itself say micro means smaller injections. So we do what? We will use small needles or the syringes or pipettes and directly put it inside that syringe into the cell wall, like the way injections are put inside the body through the, whether it's a muscular injection or it's the intravenous injections, depending upon, and then we directly inject the things. Same thing over there. Okay, so micro injection is also called as micropipette, micro petals, or glass needles, directly injecting the DNA inside the cell. Okay, then we have particle guns, which is also called as gene guns and also called as biolistic. So here what happened, tungsten with the gold coating or the gold particles, we coat it with the DNA or the gene of interest and directly do what, shoot up. So imagine that someone will shoot where the bullet will go inside me. Same thing with the cell also. So these are microscopic guns which we will directly shoot with the high velocity. It will go inside and bombard inside and goes inside the cell. Okay, so this is what, tungsten or gold particles coated with foreign DNA are bombarded into target cell. Then we have liposomes. Liposomes are what? They are the lipid bags. Name itself saying that they are the lipid bags. They are fused directly with the protoplast. Okay, are fused with protoplast. With the cell, we directly fuse it up, and that's how our this thing, fifth step, is over. Direct and indirect ways. The next steps would be what? The next would be the that you have put it inside it. Now you have to find it whether the one has the genes or not even the genes, or whether it has finally uptaken it and will show the effect or not. And the same thing happens what, that in a classroom, and if someone has like diaries has been given to every student, the next day class teacher will come and check that whether everyone has the diary or someone has thrown it away. So same thing like that. Okay, so we have to find those students who have thrown it away because they are of no use to us. Clear? So comes our sixth step, that is selection of transformant. Transformant, those who have those cells which has taken up the DNA. Okay, selection of transformant with recombinant DNA. Okay, So here we're gonna be employing two different way of transformation. Number one is on the basis of PBR322 plasmid, where the antibiotic resistance genes were there, and the other one is for the LAC-Z gene. Okay, let's try to understand what is happening over there. Imagine that I have a capability, okay, and that is because of a gene which is giving me capability that I can survive in carbon dioxide rich environment. Do you think that anyone can survive in carbon dioxide rich environment? No. Like if you put me on a Mars, I will die. I need an oxygen mask so that high pressure of oxygen is there, because I can survive in that only. But somehow I have the capability that I can survive in carbon dioxide environment. So what scientists did, they took me and throwed me on the Mars. But on Mars, there was an alien who wanted to do an experiment on me. And what it does, he want to add me for the nitrogen rich environment. So he took the genes over there and put where there was carbon dioxide rich gene present. So he cut the gene, he put the nitrogen gene in place of that, and then he put me and I died. Why I died? Because outside on the Mars, the environment was what? Carbon dioxide rich. I have lost the superpower of being able to resist in the carbon dioxide. And that is what called as what? Inactivation. Means my feature has been inactivated. But why? What? By inserting something new. So this is called as insertional inactivation, and this is only the concepts we're gonna be taking up to find out the transformant from the non-transformant. Means the transformant which will have the new gene, it will lose the capacity to survive on the carbon dioxide rich environment. Understood? Let's just see that. So what happened, that what we did, let's ligate the foreign DNA at any of the one antibiotic site. Let's take the BAM H1 endonuclease we have. We will cut the site of the BAM H1 endonuclease, okay, of foreign DNA at BAM H1 site of tetracycline, because tetracycline will be cut over there. Tetracycline resistant gene in vector PBR322. I just wrote it wrong. That is PBR32. Now what will happen? The recombinant plasmid will lose tetracycline resistance gene, because I have made it a cut. Once you have made it a cut, then the bond is not continuous. So continuous formation of protein won't occur. Feature is lost. Okay. Resistance due to insertion of foreign DNA. It is called as insertional inactivation. Now it can be selected out from non-recombinant, because there will be some of them like that extremely stubborn bacteria who has not taken that one. We can find it out. Okay, recombinant ones by following basis. So what will happen? Let's put the scenario. You will tell me that what gonna happen. Okay, so first case, what we are taking, if we put the recombinant ones, means which has that recombinant on ampicillin, there is no talk with the ampicillin. Ampicillin will, no one is cutting it, no one is doing anything with it. Plus we are putting the non-recombinant also, the ones who is not taking anything. So recombinant and non-recombinant on ampicillin, they both will grow or not? They both will grow. Why they will grow? Because we have done nothing with the ampicillin. Means both the category of the antibiotics, recombinant, non-recombinant, they are able to survive. Okay, survive and grow. As nothing has been done, okay, no inactivation has been done. Let's come to the second option. The second option is that recombinant, means where we have added like the ones which are cut and the insertion has been taken place. So recombinant on tetracycline means those who are have this DNA, they will what? They will die. No growth. Why? Because the feature capability of resistance to it, means the capability of my on a carbon dioxide is lost. So they have also lost. Means the recombinant, once you do it, something you will do some poking things to them, they will lose it. Okay. Whereas the non-recombinant means which do not have the gene of interest on tetracycline, means because they have still might have the cycle present, might still might have the feature present, so they will survive and grow. So with this fact, what we come to know that? So we come to know that the non-recombinant, so the non-recombinant will grow on both. So the reason would be why they are growing, because the case would be that once the plasmid that was there, we have opened it up, but plasmid can again itself close now, because plasmid when we are cutting up, what is happening? Its sticky ends are still available now. So one option is there, it can self ligate also. Okay, so one of the reason could be due to self ligation of plasmid. Okay, moving on to the next is, in this way, one antibiotic resistant gene helps in selecting the transformants. So this is a one way of it, by using what? By two antibiotic genes. This is the first way of doing it. What is the another way? Another way is like, what is the problem with this way? In this, the major problem is that the gene that you are like, for example, this is your first plate. Okay, let's take an example. What's the problem? This is the first plate, second plate. Okay, so this we have plating. So we are doing what? We are putting the bacteria into it. So in that first case, this is one colony, and this is, let's take the another colony. We saw that we put into two different medium, and this is what the growth is look like. A colony, B colony. So what basically happened? So first I put it, step one, I put this is on tetracycline. So this is what it is filled with ampicillin. So in ampicillin, what is happening? Both is growing, both survives. But when I put it on the tetracycline, both of them, then what happened? Only A survives and B dies. And I am telling that these are the ones which are what? They are the ones which contain the recombinant one. So those which dies, which are not present, they are what? They are by recombinant. Because here what happened? Insertional inactivation of this gene took place. Okay, so what I will do? So in the first plate, this B1, I will take it out. I will take it out and go for the next process. Understood? So this is the major question, that sometimes it can occur that when we are actually finding which are the recombinant and non-recombinant, first we put, like, this is the Petra dish, I put the ampicillin, both survive. Okay, now I put the tetracycline, the recombinant dies and they survives. Which means what, if they are died, that how I will make them do my work, whatever the work, recombinant work I want to do, because of the gene that will present, the cells have died, how I will do it. So actually what happened, we use a Ledberg's method of imprinting the bacterial colony. So what we do, this first step 1 and step 2, the step 2, before to go into step 2, we make what, replica of the first colony. So this is the first, our Petra dish, we put a velvet cloth and we take the clog on the another one. So this is what, where we are doing the experiments, but we have a replica copy over there. So wherever they will die, we will find it out and we will take that thing. I hope this thing is understood. A very important point to remember. So here we have, here the second way of is what, the major problem. So here what it is, that is selection of, okay, here we will write, they are the recombinant ones. So selection of recombinant due to inactivation of antibiotic resistance gene. We are just writing the facts, nothing much. Okay, antibiotic resistance gene is a cumbersome process. Why? Two plating we have to done. Okay, procedure, because it requires simultaneous plating on two plates having different solution, means antibiotic, different solution antibiotic. So alternative to selectable marker of antibiotic is the LAC-Z genes, which is basically what it talks about, that if that is present, it will give a blue color substrate. If we will cut it and if we give the bacteria the basic working which is it's doing, it won't be able to produce that blue color colonies. And we are able to find out what? We are able to find that, okay, these are the recombinant ones because they are not producing the blue color colonies. Easy peasy, no simultaneously plating on any one of it. Okay, so marker have been developed which differentiate recombinant on basis of their ability to produce color. So what are they? So what desired gene is inserted within the coding sequence of an enzyme which produce color. Okay, enzyme. This is what we are doing. Insertional inactivation enzyme for chromogenic substrate Z girl gives blue color. Okay, maybe like this. Now this This gives blue colored colonies if plasmid in bacteria does not have an insert. Means desired DNA absent, because that has not taken the ligation thing. Okay, so now presence of insert will do what? Insertional inactivation. And what will lead to it? Colonies does not produce any color, means they will appear what? Transparent or white. So these will be what? The recombinant colonies. Okay, so they will be what? Thus recombinant colonies. Now once we are done with it, the next step is the next seventh step. Okay, now the seventh step said what? It is that you know that these are the recombinant one. Now you want it to make your product. So we're gonna be doing with that step, that is obtaining the foreign gene products. Now let's see. In all recombinant technologies, ultimate aim is to produce a desirable protein. And hence there is a need to form it on the large scale. Hence there is a need for recombinant DNA to express this feature. Or produce this feature probably. Okay, so after having cloned the gene of interest and having optimum or optimized conditions to induce the expression of the target protein, one has to consider producing it on a large scale. And to produce it on a large scale, we need bioreactors. But before coming onto that topic, we had to understand that what these bioreactors are actually doing. They are keep on increasing their population and keeping the whole thing in their log phase. What is the log phase? Let's try to understand. First, let's write it up and then I will explain you all that thing. And here comes the most important. In large biomes or biomass culture, just cells is multiplied continuously, in which used medium is drained out from one side while fresh medium means on which the cell will grow. Okay, the fresh medium is added from other side to maintain, because you know that someone has to be in the log phase continuously in growth and exponential phase. They need the basic necessity, they need food, they need resources, they need shelter. So we have to do whatever the waste and all those things are being produced, or the product that has been produced, keep on taking it out and keep on adding the fresh food, water and everything, so that it can sustain and keep on going. This is how we are doing exponential growth in terms of human population also. That currently we have we have space, we have food, we have so many things on the planet earth, that we are exponentially reproducing, but at a certain rate it will be restricted. Okay, to maintain the cells in their physiologically most active log, or the exponential growth phase. So means what? If this is your graph, then the growth would be like this and it will be stable. This is a realistic growth. So this is representing what? Realistic growth. This will be what? This is a carrying capacity. We will learn about it. This is the maximum it can carry. After that, the growth will be stagnant. Not much growth will be there. So we want this phase when there is what? Small. This is the time. And this is the population. So see how much population grows in this small period of a time. Can you see this? That in this period of a time, so much population is grown, this much population has been grown. So we always want the whole situation to stay in this. Okay, so this is what? This is the log phase. We want that cell stays in this phase only, where in the small period of a time they can produce high amount of proteins or the product. I hope this thing is understood. And to do so, we have what? We have bioreactors. Okay, so for this bioreactors were developed, which produce large volume of products. How much that volume could be? 100 to 1000 liters. This much product has been forming with this. Okay, let's just talk about the bioreactors. Bioreactors are the vessels in which raw material are biologically converted. Meanwhile, coming back to very important update, that is I am starting a very own one-on-one mentorship program for your board as well as NEET. This is very good way, because I know when I was in my 12th, I know that by this time when the October starts, the feeling, palpitations, the feeling of what to do, where to do, how to crack everything comes into way. And there only when I needed the most a mentor, it was not there for me. I tried to go to my teacher. They give some reviews, but they really know that what I'm going through with it? Maybe not. You need a mentor, and everyone needs a mentor. Being a teacher, I have my own mentor, and you need one mentor. Maybe I could be helpful for that. For that, you can go to my uh, Instagram page and know about all the potential courses. And I must say that it's not any kind of to the parents, this thing is there, it's not any one time payment things, it's an investment on your child for your counseling process also. Till the counseling, I will be the support will be given, even if the fees will be taken only till the March or April end. Coming back, a very important thing is that the investment part is that if your child scored 360 out of 360, then whole of the fees from the first month to the last month will be given back to you. I felt that this is the best thing available in the market for your child and for you students. So if you're looking forward, can join that. You can get all the details about it on my Instagram. Okay, that is chlorophyll_classes on the Instagram. So coming back to the major topic, that is bioreactors. They are biologically converted into specific products or individual enzymes, etc., whatever we're gonna be making with it. Using what? Using the microbial plant, means basically the microbes that will be doing it, animal or human cells, whatever, wherever we want to do it, we will make it do it. The major important point is it provides the optimal conditions for the growth of the cells. Okay, so optimal temperature conditions means what? That oxygen, shelter, water, food, everything. Okay, provide optimal condition, produce optimal condition for achieving desired products by providing optimum growth conditions. What are those growth conditions? pH, temperature, oxygen, etc. All of that thing would be coming into it. Going on the bioreactors, there are basically two types of bioreactors. Okay, so bioreactors type, then it is what? It is simple stirred type of, and another is the parched stirred. Before to made you understand this, let's see its diagram from NCRT itself. Okay, so I'm going to be adding a diagram from NCRT. So these are the ones. This is the simple stirrer. And this one is the other one around. So can you see the first one, the simple stirrer? In the simple stirrer, what is happening? There is what this kind of thing is there, and there are blades present. Just like your mixie or the mixing juicer or something like that. So it is keep on stirring what? Circulating the substances. So that substance don't keep on settle down. So that the bacteria which is present above, it might not be getting the things, but what it does, it's keep on stirring the whole batch, keep on like this. So it happens what? Everything is circulated to every corner. Okay, then the second thing is, if you see that over here there is what, this pipe, this is what stream for sterilization, because continuously there might be what, if you are adding certain things, there might be possibility that microbial infection occur. If the infection will occur, what will happen, it will destroy your product quality. So we have that also, we are adding up. We have acid base pH control also, that if what is the needed can be given, because organism will live in a particular osmolarity condition. Then we have a motor, we have foam maker. So if the foam is here, we want to break it up, it won't be there, because it will do what, it will deplete the content of the supply of the things. Then these culture broth is there, which is written over there, flat plated embeller is there, and sterilized air is also being added. Air will be keep on adding through the bottom, so that it can goes to the up. Okay, and then what is that, they have the sparge. If you see over here, there is also one thing is there which can keep on moving, but here one extra thing that is happening is what, that there is a gas that is being formed. Okay, and that is what, that this is present over here. Okay, the gases and the gas bubbles and all those things, which is an extra way of providing what, aerobic conditions if needed. So I'm going to be taking about that. If you see the other way around, that this is what, this is a flat base, it is a curved base one. So this is also one of the difference. So I'm going to be writing these things on the next page about it. Okay, so simultaneously I'm writing it up. Okay, number one is, in simple, it is what, cylindrical in shape, or having a curved base, having a curved base that simplifies the mixing, that simplify the mixing of the reactor, that simplify the mixing of reactor substances. Now the stirrer helps in uniform mixing and availability of oxygen throughout the bioreactor, throughout the bioreactors. That thing we have already studied. Here what is the different and the important point is that the sterile air is sparged throughout the reactor, and has increases surface for oxygen transfer. Then simple stirrer. That's the major thing. This is the major thing. The surface area is more, which means what? It means more aerobic respiration probabilities and things would be there. Now, if someone will ask, what are the advantages of this? Okay, so the next thing is advantages of bioreactors. So it goes like this, that first of all, it is important for continuous operation. It keeps on doing the continuous operation. We don't have to keep on think about anything, that how, if the operation is stopped, whether it will stop, what will happen. No, it will keep on happening. Then it has good temperature control, it has good control over the process. Then low operating cost, because if humans will be there, multiple steps would be there, then it will, what, cost would be more. So okay, so low operational cost is there, which makes it what, suitable. Now once we get it, can we directly give the things to the humans? No, we have to run the clinical test, we have to put the expiry date, we have to check that whether it is of right quantity or not. So that is the last step over it, that is downstreaming process, simplest which have I explained you. Let's just write about it. Okay, so eighth is what, downstreaming or downstream process. or processing. So now in this, after completion, after completing of biosynthesis with the help of reactor, biosynthesis stage, the product is subjected through is subjected through a series of processes, before it is ready for marketing as a finished product. Marketing as a finished product. So it includes separation and purification, more products has to formulate with preservatives, because we don't want it to be waste before even reaching to the consumers. Okay, that would be a loss. Okay, has to undergo clinical trials, means whether it is suitable for the human consumption or not. We will try it on the monkeys and then on the humans. Okay, strict quality control, strict quality control testing before releasing into masses. Before releasing into masses. That's about the whole process. And with this, our chapter is also over. If you like my videos, give it a like, share it with your friends. I even come up with a course whose details you can get it on my Instagram page, that is chlorophyll_classes. All the traits, how effective it would be for your boards also, for the NEET also. So there are two different courses we are launching for the boards, for the NEET, which you can definitely check over on my Instagrams. And don't worry about the fees, it's a very affordable one. Go check it out yourself. Well, that's all for this video, and I taken over. Thank you.